So, hi everyone, welcome to another talk in of Fishes. Today we have with us Dr. Oliver Meacock, who is a research fellow in the School of Biosciences at the University of Sheffield. Today he is going to speak about how microbial communities interact with each other, not only through themselves, but also through environments, and how there are some interactions which emerge based on population scales. I'll hand over to Oliver.
Okay, thank you very much. So as I was saying to people earlier, um, please do ask any questions during this talk that you like. I'll try and keep an eye out if there are any hands up during the talk. Uh, I've got 45 slides and 45 minutes, so that should be about the right number of each. But if we don't get to the end of them, it doesn't matter. I think it's better to have a more in-depth discussion than to have really just every piece of information supplied and you not understand it. So I'm going to be talking today about, as was introduced, microbial ecosystems and in particular, how we can use ecological theories to understand them. So as some of you might know already, microbial communities are not only everywhere, they do lots of different things. So, for example, they have important roles in soil function and health. They also have important roles, obviously, in the gut microbiome. And for biosynthetic purposes, we might be interested in constructing synthetic communities that can build molecules that we are interested in. So, if we want to introduce species, or if we want to exclude species, or if we want to build communities that stably coexist together in order to perform these biosynthetic functions, these are really ecological questions that we're asking. So how is it that we can get a species to invade stably? Or how is it that we can drive a species to extinction? Or how is it that we can maintain stable coexistence of multiple populations simultaneously? So given that these are ecological questions, it's natural that we as microbial ecologists should have turned to classical ecological theories in order to explore these problems. So there's two big kinds of threads of thought that have been brought to bear on this problem. The first of these frameworks is what I'll call the phenomenological framework. So that is where we have systems of this structure, where we represent per capita growth rates as some function of the population densities. And these are just represented in this vector down here. So we can have lots of different populations in our system. We can have big interaction networks of lots of different populations interacting with each other through this kind of relationship. And the key idea here is that we've got this density-dependent relationship between population density and population growth rates. So, for example, if we had one species growing by itself, we might expect to see this kind of relationship, where as that population becomes more and more dense, the growth rate decreases. And that is indicative of what we would call a negative interaction of that population with itself. So as you increase the density, the growth rate comes down. Under other circumstances, and particularly if you've got two species, you might see that this relationship is the other way around. So as you increase the population density, you might see that the growth rate increases. And that's what we would call a positive interaction in this kind of framework. Now, the most common of these phenomenological models is what's called the generalized Lotka-Volterra model. And this is basically just a model where we assume that this relationship down here is linear. But there has been interest in recent years of alternative models. So this was a paper which caused a little bit of a splash a couple of years ago, where basically they asked what happens if you've got non-linear relationships here, so sublinear growth. But today I'll mostly be talking about this generalized Lotka-Volterra type of viewpoint. The second perspective is to really focus on the mechanisms through which populations interact. This is particularly relevant for microbial ecosystems. So now instead of just representing our population densities, now we have also this vector which represents all the quantities in the environment. So this might be, for example, quantities of resources, which is mostly what I'll be talking about today, but it could also be for example, pH or light availability or water availability in a plant model, for example. So this is a very general kind of view of the environment. And the key idea here is that we're splitting the world into these two compartments, the environment and the populations, and these two compartments can interact with each other, but they don't interact within themselves. These models have this kind of general form, which I will come back to later and explain more thoroughly, but there's lots of different studies out there that use this kind of approach. So given that we have these two different frameworks, it's natural to ask what the relationship is between them. So we've got these mechanistic models where we have this explicit representation of the interaction between organisms and their environment, and then how that leads to effective interactions between the populations. But then we also have this phenomenological model over here, where these processes have effectively been abstracted out. So we've lost some resolution, but in principle, we should be representing the same processes. And both of these frameworks have relative advantages and disadvantages. So by putting realistic constraints on mechanistic processes, these are generally much more effective at recapitulating population dynamics in a kind of fine-grained detail, but they're hard to generalize, and you also need to make lots of measurements of the environment in order to parameterize them. And this is very painful for microbiome studies, for example, where the number of factors through which species could be interacting. So these could be metabolites in your gut, for example. These can be numbering, 20,000 different quantities. So if you're trying to build a model based on 20,000 free variables, that's going to be very difficult. Over here, in these phenomenological models, they're great because they're relatively easy to parameterize. You just need to measure these pairwise interactions between your populations, capturing that density-dependent regulation. But on the other hand, there's lots of phenomena that these models don't capture, and that's basically because we've lost this mechanistic detail. And also these models generate paradoxes that seem to contradict what we see in nature. And I'll give some concrete examples later on of those paradoxes. So a natural question to ask is, what is the connection between these two modeling frameworks? And this is a very obvious question to ask. It's something people have been asking for at least 50 years, but there's not really been a very concrete answer to it. There have been specific examples, but we've not really been able to do anything kind of generally. So what I'll be talking about today is, in the first part, I'll be describing some new results which provide us with this connection. So it's a very flexible way of moving between these two perspectives. And I see this really as a kind of Rosetta Stone for this problem. So you can basically take a mechanistic model and then you can translate it into Lotka-Volterra terms effectively, and then see how those Lotka-Volterra terms are influenced by the mechanisms. And now that we've got this Rosetta Stone, I really feel like an Egyptologist in 1800 or something. We just discovered this way to translate hieroglyphs, for example. And now there are all these phenomena that we found in ecology over the last 100 years, predominantly thinking in phenomenological terms, but now we can start to explore them explicitly in mechanistic terms and see how they kind of relate to underlying interaction mechanisms. So I'll be talking about these three today. So context dependencies, modern coexistence theory, and the cooperation stability paradox. But just to say, there's lots of other directions that this could potentially go in the future. All right, so I will just now explain my formalism before we really get into the nitty-gritty of this. So what I will be doing is showing you relationships between effector species, which I'll denote with this index beta, and then this target species, which I'll denote with this index alpha. And they'll be interacting through these intermediate compounds, which I'll denote with rho. So these are just indices. I may also use different labels at various times. And these different quantities, as I said before, they're stored in these density vectors and environment vectors. So the effector species can influence the environment through these impact functions, which I'll represent in blue. These are effectively telling you whether the intermediate compounds are secreted, which I'll denote with arrows, or whether they're depleted. So this might be uptake or it might be degradation. And then we have the sensitivity functions, which I'll represent in purple, which indicate whether those intermediate compounds increase the growth rate of the target species or decrease the growth of the target species. And if you've ever seen a consumer resource model, this is basically the same thing. We just refer to them as environment organism models because the resources don't necessarily need to be resources. They could be toxins, for example, and the species don't necessarily need to consume them. So there can be lots of like the fact that you've got multiple different signs going on here. It's basically a generalization of that consumer resource idea, but very similar concepts in play. So this is just a representation of these environment organism models, again, in a slightly more holistic way. And so you can see here that we've got these equations which define the dynamics of these systems. So we've got the environmental dynamics, which are determined by these impact functions, remember, and we've scaled them by the population densities. But then we also have what I'll call this allergenic function, and this is just external influences on the environment. So the environment can be manipulated both by the organisms inside the environment, or there can be external fluxes into that environment. So this could be, for example, if we've got food coming into an ecosystem externally. And then we have this purple arrow over here. These are the sensitivity functions, which just describe how the per capita growth rate varies as a function of the environmental state. And I'll be comparing this to these phenomenological models, which remember, we just have this direct density dependence. And I'll be focusing on this generalized Locker-Volterra model, where we again have this per capita growth rate, but that is defined in terms of these constant intrinsic growth rates, mu alpha. And then we have these interaction strengths over here. So this is these linear density dependent values. So if you see that these interaction strengths are positive, that's what is usually called a cooperative interaction. If they're negative, that's what we would call a competitive interaction, or they can be zero, in which case we would call it a neutral interaction. Right. And so so if we want to kind of understand this connection between these two different frameworks, it would be very useful to be able to understand how we can write this type of system in these terms. So is there a way, basically, that we can combine these equations together in order to get parameters that look like these Locker-Volterra parameters? Again, that's a kind of obvious question to ask, and people have also been doing that for a long time. So MacArthur did this in 1970. He basically showed that for one specific model of this type, you can do some algebra, combine your components together, and you can derive something that ends up looking like Locker-Volterra. And that gives you then your understanding of how those interaction mechanisms give rise to these processes. But there's something a little bit challenging with that, because if you look over here, what we have is two different timescales, but over here what we've got is a single timescale. Now the trick that Robert MacArthur used was that he eliminated one of these two timescales. So he basically said the environment equilibrates a lot more quickly than the organisms, and so this timescale is going to disappear. And then we're going to have a rate-based equation which is going to map on nicely onto this. But you can show that this only works for this one particular model. So what we asked is whether or not there is a way that you can avoid that assumption and basically combine these two timescales together. And in doing that, what we're going to arrive at is a definition of interactions, which is mediated via these two sets of processes simultaneously. So I'll just be a little bit more explicit about what I mean here. So we've got our sensitivity functions here and our impact functions here in blue and purple. And what I'm representing here are different interaction mechanisms through which populations can interact. So we can have secretion of nutrients. So beta can be secreting a compound which is of use to alpha, for example. That's what we would call enrichment or cross-feeding. We can also have detoxification. So if this is a toxin down here, you can have suppression of that toxin. Both of these are positive interaction mechanisms. And then we have negative interactions mechanisms here on this diagonal. So this compound might be a nutrient, but it's being depleted by beta. So that would be a negative effect on the growth rate of alpha, or we can have production of toxins, which suppress the growth of alpha again. And these mechanisms can be stacked on top of each other in order to give us more complex interaction types. So we can combine, for example, enrichment and detoxification, both of these mechanisms have a positive effect on the growth rate of alpha, and so the net effect of both of those together is positive. We have a very similar idea here where we've got depletion and pollution occurring simultaneously. Both of these have negative effects on the growth rate of alpha, and so we would call this a negative growth rate effect. And then over here, what I've done is I've combined together two mechanisms that have opposite signs. So we've got this depletion, which is competition for resources, but then we also have down here detoxification, which is a positive effect. So this might be, for example, removing an antibiotic from the environment. And in this case, what we end up finding is that the net effect of beta on alpha will depend on the relative concentration of these two different compounds. So you might find that in some circumstances you're going to have depletion dominating, and in other circumstances you're going to have detoxification dominating. And so this is kind of a more subtle idea of, sorry, I'm just going to put the laser pointer on. So this is a more subtle idea of what an interaction is. It's a net effect of multiple interaction mechanisms occurring simultaneously. All right. So let's go back to this picture. So I said before that I'm going to combine these two processes together, and then that's going to give us, in some sense, an interaction, which is then going to map onto this interaction concept in the phenomenological general Locke-Volterra model. So how does this work exactly? Well, this is just a little bit of algebra, basically. So we start off with our sensitivity functions defined over here. So just remember, this is just defining the per capita growth rate as a function of the environmental state. And now I'm going to take the derivative of these sensitivity functions with respect to time. So this is this idea of combining these two timescales together. We're not eliminating one of them by assuming that we've got fast dynamics of one of the compartments. Instead, what we're doing is we're allowing both of those timescales to occur simultaneously. When we do this, we take the temporal derivative of our growth rates, and we perform the multivariate chain rule on this, and this splits this term into two different components. So we've got the sensitivity gradients over here. So these are the gradients with respect to the environmental factors. And again, I'll give a concrete example of what this looks like in a future slide. And then we've got these environmental dynamics over here. Now we've got an equation for the environmental dynamics. We've seen that before, so we can just substitute that in here and then perform a little bit of manipulation. We obtain this equation down here. And then what I'm going to do is I'm going to call this term over here the abiotic accelerations, and I'm going to call this mu prime alpha, and then I'm going to call this term over here the biotic accelerations, and I'll call that a prime alpha beta. All right, so these abiotic accelerations, they tell you how quickly the environment is changing the growth rate of alpha. And these biotic accelerations tell you in per capita terms how quickly the other organisms in the system are changing the growth rate of alpha. All right. And I've written this somewhat suggestively, and there's a very good reason for that, because you'll see that these two equations do look quite similar to one another once we've expressed them in this way. So I want to emphasize that this is not Locke-Volterra. So we have a second order equation now, accelerations rather than rates, because we've combined these two timescales together. Whereas over here, what we've got is a rate-based first order equation. Also, these effective parameters are expressed in terms of these environmentally dependent quantities. So these can change over time as the environment changes, for example, or in different environmental contexts. And we'll see again this in the future. However, it's still nice to be able to express them in this way because at equilibrium, when both of these sides are equal to 0, these two expressions end up being algebraically equivalent. So we end up having some sort of equilibrium environment, R star, and these effective Locke-Volterra parameters then become algebraically equivalent to these terms over here. Now this turns out to be extremely useful because it means that if you've got an expression which is true for Locke-Volterra at equilibrium, and there are lots of these around, lots of theorems, lots of inequalities, for example, you can understand, what this result tells you is that you can turn this into an expression which is true for every environment organism system, merely by replacing any rates that you see over here with accelerations over here. And again, I'll give you an example of what that means later on. But basically, this turns out to be very useful for lots of reasons, and this is going to be what the kind of Rosetta Stone idea is in this talk. So this is the symmetry which allows us to translate between these two different frameworks. And just a final slide on this introductory section, just to again re-emphasize this idea of an interaction being mediated via different mechanisms. So as I was describing before, we've got this net interaction, which is a combination of multiple different mechanisms occurring simultaneously. If we look at this scalar product down here, that's exactly what's represented by this biotic acceleration. So we've got the growth rate impact of each individual mechanism, which is the product of how much that change to the environment changes the growth rate of the focal species, combined with how much the effector is changing the environment. So that combination is then multiplied together and then added up, and then that gives us this biotic acceleration. So this is quite nice that this kind of intuitive idea of what a net interaction should look like ends up just popping out of the algebra directly. Now, I will sometimes slip into calling these interactions during this talk. That's not technically true. Do bear in mind that these are not necessarily the same as Lotka-Volterra interactions, and we'll see some examples where these do not behave at all like Lotka-Volterra interactions, in fact. But nonetheless, it's still useful to think of this in a kind of interaction term. And it's useful to keep that kind of semantic distinction between Lotka-Volterra interaction strengths and these biotic accelerations. All right, so moving on to the applications now. So I'm going to start by asking why it is that interactions are context dependent and whether or not this framework can help us to understand these context dependencies. So firstly, what are context dependencies? So if we think about these Lotka-Volterra models, we have these fixed interaction strengths which represent the growth rate impact of different populations on one another. And what people have been discovering in recent years is that actually these don't seem to be fixed. So this is a meta-analysis of a lot of different studies that basically tried to measure these interactions between different populations. And they found that depending on the context in which these interactions were being measured, so this might be in different communities, or it might be in different environments, or lots of different contexts, you find that the sign of the interaction can actually change. So you might go from being a positive interaction, for example, to being competitive, or positive to neutral. So and don't worry too much about what these different bars represent. Basically, the key point here is that you see this is very common and very widespread. So if you're interested in this from a microbial context, Switzerland is a very useful place to be doing a postdoc because a lot of the work has been doing, a lot of the work in this area has been coming out of Switzerland. So in the field, in the lab that I was in with Sarah Mitri, they had this nice study where they were investigating how cells grow in toxic environments. And basically what they found was that as you increase the concentration of toxins in the environment, your interactions become more and more cooperative. So this is related to something called the stress gradient hypothesis, which basically says as the stress in your environment increases, you expect cooperation to become stronger. And that's what they found in this study. This is work from the group of Martin Ackermann, where they basically asked how interactions change as a function of time. And you can see that, again, interaction strength can change from being neutral to negative in this purple case. And then we have this third example, which I will mention briefly, but won't talk about too much, which is where we have spatial dependencies. So these are microfluidic devices where basically you've got unidirectional flow in the system. And you can see that there's this orange species over here, which can only grow downstream of the green species. So you would say that there's a neutral interaction maybe between the two populations up here, but then we have a positive interaction from the green species to the orange species that gets increasingly strong as we move along the length of the channel. So can we use the equation that I showed you before, this second order accelerational equation, in order to understand these processes? So as I mentioned before, these effective Lotka-Volterra terms, they are dependent on the environmental state. So maybe we would expect that we can capture some sort of environmental dependency through them. So I'm going to start with what I will call the kind of hydrogen atom of these environment organism models, which is where we close off the system from external inputs. So this is where we've set the allergenic or external processes to 0. So we do this just by growing our cells in a flask, basically, so nothing's allowed in or out. And then I'm just going to consider a single population growing by itself. And we're going to ask how that population is interacting with itself. So this is kind of as simple as it gets, but it's a useful way of understanding how all these different concepts kind of interact with each other. So this is going to be our model system. So we've got this population A, which is interacting with itself via depletion of this nutrient and detoxification of this toxin down here. And these are the explicit representations of these impact functions and sensitivity functions. So our impact functions just represent how the environment is being pulled by the population over time. So you see that because this population is reducing the concentration of both the nutrient and the toxin, we see that these arrows tend to point down towards the origin. And then we've got this sensitivity function over here, which represents essentially the per capita growth rate as a function of the environmental state. And so you can see that that's negative up here where we've got lots of toxins, but positive down here where we've got lots of nutrients and not very many toxins. Now this sensitivity gradient is represented with these black arrows, and you see that these tend to point in the direction where the growth rate is increasing the quickest as we vary the environmental state. So basically what this is saying is in which direction does this population want to be pulled in order to grow as quickly as possible. So when you combine these two vector fields together, this field tells you how quickly the environment is being pulled in each direction, and this tells you how quickly it wants the environment to be pulled in order to increase its growth rate. So if those two arrows align with each other, you'll get a positive value, and if they point in opposite directions, they will give you a negative value. And that's what's represented over here. We've got this scalar field, which represents these biotic accelerations, which are these interaction-like quantities. So in some environments, we find that this detoxification mechanism dominates over here, but in other environments, we find that this depletion mechanism dominates. And so you can see generally, you see detoxification needs some toxin in the environment, and it also needs some nutrients. Okay, so that gives us a kind of static picture of how the environment determines these interaction values. But remember that we're in a closed batch culture system. So in these systems, what you do is you put in all your nutrients and you put in all your toxins at the beginning, but the populations will change those concentrations over time. And so what you're going to have is a trajectory in this environment space. So we start off with a certain concentration of our nutrients and our toxins, but then both of these get depleted, and then we move down towards the origin as that occurs. Now you'll see in this plot, we're moving from a region where we've got positive interactions down to a region where we've got negative interaction, or I should say biotic accelerations. So what we might expect to see is that if we were to measure some sort of effective interaction value, we might expect to see that switch in sign from positive to negative. So that's kind of what they were capturing in that meta review that I mentioned earlier. And just to say, we can have lots of different starting environmental states, and we're going to have lots of trajectories in this. So this would be, in this case, 16 different batch culture experiments where we've started each one at a different position in this environment space. And then we're going to see how do the dynamics change the interaction value. So we can realize this experimentally. So this is a soil bacterium called Coemonas testosteroni, and it turns out that it competes with itself over proline, but it also degrades ampicillin, which is an antibiotic. And we can inoculate this population into batch culture systems which contain different starting concentrations of proline or ampicillin. So that's effectively what we were representing in the previous slide. And then we can ask how the interaction between this population and itself varies as a function of time. So if we start in the case where we have no ampicillin at all, so no toxin in the environment at all, we find that there's no positive interaction phase. And that makes a lot of sense. We don't have any toxin, and so there's no way for that positive interaction mechanism to actually manifest itself. So remember that in order to see a positive interaction, we need to have some toxin to be degraded in order to see a positive effect. However, as we increase the ampicillin concentration and keep the proline concentration fixed, we find that there is a positive interaction phase, and that the strength of that positive interaction phase increases as we increase the toxin concentration. So we have both this environmental dependency, where we're ramping up the toxin concentration and seeing that the positive interaction phase becomes more positive, but then we also have this temporal dependency, where the interaction changes sign over time and becomes negative by the end of the experiment. Now we can summarize these experiments just by measuring the most positive and the most negative interaction values, and we can compare between the experiments and the model, and we find that there's a very nice correspondence between these two. Now, these are not quantitative results, so I've not fitted this model to this experiment, so think of this as a qualitative kind of framework, but nonetheless, it is nice that we are able to reproduce these context dependencies through the model. Now, as a final point, which I won't dwell on for too long, we can, as I say, also bring spatial dependencies into this framework. So let's look at this microfluidic device again. And we're also going to consider a slightly more complex community where we've got two populations and we've got multiple different interaction mechanisms occurring again, as before. Now, when you've got two populations, instead of just having one of these environment spaces representing the interaction of this population with itself, now we've got scalar fields which represent interactions of these populations with each other. So that's these off-diagonal components, as well as the on-diagonal components which represent the interactions with themselves. So interspecific interactions, but also intraspecific interactions on this diagonal. And there's a nice mathematical correspondence which you can use to show that in both these batch culture systems and these spatially dependent systems, you can use exactly the same mathematical machinery in order to describe the context dependencies in this system. I won't go into detail. If you're interested, read the paper, but it's just quite a cool mathematical symmetry there. All right, so I'll move on to the second application, which is to ask why microbial mutualisms are stable. So this is the preprint, which I think you originally got in touch with me about. So this is more recent results. So you might be wondering why I'm asking why microbial mutualisms might be stable. I mean, what is a mutualism to begin with? Again, you might not be familiar with these. So basically, these are any relationship where we've got a positive association between two different populations. So you look out of your window, you'll probably see some bees or some butterflies or something like that are pollinating flowers. That's a classic example of a mutualism. Both populations benefit. You can also have these cleanerfish and their clients, ants and aphids. And in the microbial context, as we'll see, there's these cross-feeding exchanges between populations where one species secretes something that the other population needs and vice versa. But when we look at this from a phenomenological perspective, there's some weird results that we get out of this. So this is a kind of classic pathology in these phenomenological models, which is called mutualistic blow-up. So basically what this says is that if your two populations benefit each other too much, they're just going to spiral off to infinity. So basically you're going to have a positive feedback loop, and then the populations are just going to get bigger and bigger and bigger without bound. Obviously that's not feasible. And it's interesting to note that even in this original publication where they showed this mutualistic blow-up, they noted that there would eventually be some limitation to the system overall. I'll come back to this kind of idea later, but interesting to note that this was already being thought of in 1935. You can also have similar effects in big network models. So here we've just got two species, but if we've got an arbitrary number of species, we have very similar effects, whereas we increase the fraction of these cooperative interactions within these networks, we find that at some point the stability of the system drops off and we start having things going extinct. This is a slightly more subtle effect than what we have with mutualistic blow-up because we don't necessarily need to have divergence here. It can be that our populations end up equilibrating at finite abundances. But what those positive feedback loops are doing is stopping the system from being stable in the initial state. So you're going to end up losing some of your cooperative species. They're going to go extinct, and you're going to have a subcommunity of your starting community, which is stable in that case. You can, of course, also have divergence in these systems. So given that we see that there's these cross-feeding exchanges all over the place, how is it that we can reconcile the stability of these microbial cross-feeding exchanges with that instability of the mutualistic phenomenological models? So if you actually go and look at interaction mechanisms in the soil, you can find that over 50% of the species are actually sharing resources between each other. And these can be essential processes. So if this population can't produce a particular amino acid, for example, so if it's what we call an auxotroph, it needs to be fed that by a partner in order to survive and grow. So given that we see that there's this incredibly widespread cross-feeding in nature, how is it that we can reconcile that with the stability of these microbiota? There's a few ways that we can address this within the phenomenological context. Don't worry too much about the details of this slide, but basically what this is saying is there's lots of ways that you can draw these density dependence in order to reduce the likelihood of instabilities arising. So for example, you can say that the benefits of the mutualism might saturate at a particular population density, and it turns out that that will stop the system from blowing up. That's well and good, but what you're not doing here is you're not really grounding this in mechanistic terms. So these are sort of intuitions about how we might expect mechanisms to shape these phenomenological models. But what we would like to do now with this kind of framework that I've been talking about before, where we can go from mechanisms to interaction values and effective lockable Volterra parameters more generally, is we'd like to know how do those mechanisms actually shape these effective lockable Volterra parameters. So rather than just guessing, is there some way of seeing a sort of first principles way how those mechanisms shape these phenomenological parameters? So to do this, I'm going to be using this as my study system. So this is one of these cross-feeding systems that I mentioned before. Again, we have these impacts in blue and these sensitivities in purple. And in contrast to before, where we had a closed system, we now have external inputs to this. So this is what we call a chemostat, which is basically just a big tank where you've got your populations, it's stirred, and you have chemicals flowing into that system. So those will feed the populations in there. And these will reach a dynamical equilibrium where you've got continuous growth, continuous washout of your cells, and continuous washout of your chemicals as well. So we have this community that's embedded in this chemostat environment, and we've got these various processes going on. So this is our cross-feeding interactions over here. You can see that we've got secretion of these amino acids, V and W, and these are being fed to the partner. But then we also have this carbon source over here, C, over which both species are competing. And as before, we can represent all these processes as vector fields. So we have this impact function in blue, the sensitivity gradients in purple, and these allergenic processes in gold. And these allergenic processes I've not mentioned before, but relatively straightforward to understand what these vector fields are telling us. Basically, this is saying the system will get pulled towards whatever your input concentration is to this chemostat. So in this case, for example, we just have carbon in the influx, and so the system will get pulled towards carbon at a concentration of 2, which is our input concentration. So that's why you've got this kind of starburst appearance over here. So as before, we've got vector fields, and we take the scalar products, and that gives us these scalar fields now sitting in the environment space. And we've got this both for these biotic accelerations over here, as we had before, but also these abiotic accelerations over here. So this is where we've combined this allergenic function in gold, so these are the external inputs, with the sensitivity gradients. And you can see that we get some scalar field here as well. All right, so before what we had were trajectories, and we studied how these scalar fields varied as we moved through this space. Now what I'm going to do is I'm only going to focus on these equilibrium states, so these R stars over here. So this R star is the same in all of these plots, I should say. It's just because we've got multiple different scalar fields for each of the effective parameters. I'm just reproducing that R star 6 times. So what I'm going to ask is what these effective Lotka-Volterra parameters look like in these equilibrium environments over here. And then what I'm going to do is I'm going to ask how they change as a function of the mechanistic processes. So this is just a reminder basically saying I've set this to zero, this term to zero, we're at equilibrium, our environment is at R star, and then we have this equation which looks like the Lotka-Volterra equation at equilibrium. And we're going to explore how these parameters change as we vary mechanisms and then allow the system to re-equilibrate at a new R star. So if you were thinking about this intuitively, you might think to yourself, okay, so I'm increasing the secretion rate of, in this case, an amino acid. And we know that this population needs that amino acid. So I'm increasing the strength of the interaction mechanism. I'm increasing the cooperativity of that interaction mechanism. So I should expect the effective interaction strength to become more positive as a function of that secretion rate. Likewise, this is a costly process. I'm secreting an amino acid, which I can't use, and I'm spending carbon in order to generate it. So I might say, well, I would expect that, these abiotic accelerations, they look like intrinsic growth rates algebraically. So I should expect the kind of intrinsic growth rate to decrease. Maybe this is what I would predict. Or I might expect it to be constant. That's also a perfectly reasonable assumption. And again, this is the kind of intuitive approach that people have taken in the past. That's effectively what that Gauss paper was doing, was this kind of reasoning. If we actually look at what we see in the model, what we find is the opposite. So these are our biotic accelerations. Remember, these look like our interaction strengths. As we dial up this amino acid secretion rate, we find that they start off being positive, so looking mutualistic, but then they become increasingly competitive as we dial up that amino acid secretion rate. Now, this is even more surprising, which is that the abiotic accelerations, which look like intrinsic growth rates, also go in the opposite direction to what we would expect. So this would, in principle, solve this paradox. Basically, what we're seeing is that as we increase the strength of this interaction mechanism, things are becoming more competitive in Lotka-Volterra terms, even though we're increasing the strength of this cooperative mechanism. But we also have this compensation for that increased competition by this increased effective intrinsic growth rate. So the intrinsic growth rate will basically boost population sizes to compensate for the increased competition. So this can give you large population sizes that are stable, but it's completely counterintuitive that we have this relationship between this cooperative mechanism and these effective Lotka-Volterra parameters. So what's driving that counterintuitive relationship? So if we now look at these different R stars as a function of our leakage rate of this amino acid W, so over here what we've got is our R star at low leakage rates, and we can see that there's a fairly high amount of carbon, but a very low amount of amino acids. And that makes sense. We're at low population sizes. We have very inefficient conversion of carbon into amino acids. And so we should expect there to be lots of carbon left over. As we increase the production of this amino acid, we move along this contour here, and we end up in this place over here where we've got lots of amino acid, but not very much carbon. So if we now look at and try and interpret what this biotic acceleration is actually representing, well, over here we're in a carbon-limited environment where we have not very much carbon, but a lot of amino acids, and the interaction partner is consuming that carbon. So we should expect this competition over the carbon to dominate. Whereas over here, we are in an amino acid limited environment, and we know that the interaction partner secretes that amino acid. So we should expect the positive cross-feeding interaction mechanism to dominate. So that's driving the counterintuitive relationship of the biotic accelerations. Basically, as we increase the secretion rate of the amino acids, we have increasingly strong competition over the carbon. And then we have a very similar picture emerging in the abiotic accelerations. But instead of having our interaction partners, so that species beta, as we had before, now what we're going to consider is the effect of basically the chemostat. So we've got this nutrient influx, but also this nutrient efflux in the chemostat. And we see up here that we have, again, our carbon limited case. We see that our influx is a carbon source. And so when we have relatively high secretion rates, we end up finding that growth promotion through the supply of this carbon is what is driving this positive value over here. Whereas when we have this amino acid limited case over here, we have limited amount of amino acid, and we know that the efflux is reducing the concentrations of that amino acid. So it's being leaked out of the system. And so that is what's driving this negative value over here. So that's what's causing this value to be negative. And just to say that this generalizes quite nicely, so it's not just some weird artifact of being just a two population model. If we generalize this to 5 species, you can see that we have exactly the same phenomena emerging. So this is just showing you that we have stable coexistence of all of our different populations in this model as we increase the production rate of amino acids. But again, we have increasingly strong effective competition, but also increasingly positive abiotic accelerations, which resemble these intrinsic growth rates algebraically. And this corresponds nicely to what we see with experiments where we have multiple different auxotrophs growing together. So essentially, the summary of this work is saying that actually there's no contradiction here. So if we look at things from a Lotka-Volterra sort of phenomenological perspective, things look competitive. So in terms of these effective Lotka-Volterra parameters, and that's driving the stability of the system. But when we look at the interaction mechanisms, those look very cooperative. So we have very strong exchanges going on. What is mistaken is our assumption that the interaction mechanisms give rise necessarily to effective Lotka-Volterra parameters that are intuitive, right? So we have increasingly strong cooperative mechanisms, and that can drive increasingly strong competition in a Lotka-Volterra sense. And that is what is allowing us to have these stable cooperative communities. All right, so I have a third section, and I realize that we are running short of time. So I don't know if we would like to move on to questions at this point. I can very quickly just flash through a few pictures, but maybe we want to have a discussion more than we want to see more data. I don't know. What would you like to do?
I mean, if it's gonna take like 5 minutes, we usually go till like whatever 10:45 or something. So
Okay.
you can take your time, go through it, and then we'll have a discussion anyway.
Okay, great. Cool. I'll need to leave at 1/2 past at the latest, but I think that's fine. I'll have to stick around until then. Yeah, cool.
Oh, if you have limited time, then I think 10 minutes for discussion is better off.
No, no, 1/2 past. So we could have still half an hour after the
Ah,
end.
okay.
Yeah.
I'm sorry, because in for us it'll be 1/2 past in like
Oh,
10.
yes, sorry. Yes,
Apologies.
IST. Okay, great. Cool. Okay, so I'll go through this a bit more carefully then. So, um, the last section is about this framework called modern coexistence theory. This is a really big field of study. It was very big about 10 years ago, 20 years ago, something like that. People got very excited about it. There's a whole section of modern coexistence theory that I'm not going to mention, which is all to do with things called storage effects and fluctuation dependent coexistence and blah, blah, blah. I'm not going to mention that. What I'm going to focus on is this plot here. So this really summarizes the key idea of one of the parts of modern coexistence theory, which is that coexistence can be entirely determined by the combination of these two quantities called niche overlap and the fitness ratio. So you can have very diverse interaction mechanisms, lots of stuff going on in terms of your processes, but you've got these two numbers which ultimately determine coexistence, which are downstream of those interaction processes. So what we can do now is we can say, okay, so we've got our interaction mechanisms, and then we've got our effective Lotka-Volterra interactions, and then what we're going to ask is, how does that affect coexistence in the system? And we're going to do that using this modern coexistence framework. So what does this actually represent concretely? So this niche overlap metric is basically telling you how two organisms are competing for different resources. That's how we classically think about this niche overlap. So here are two of Darwin's finches, for example. We can see that they've got two different food sources. So this one eats nuts and this one eats flies. And so the niche overlap is 0 because they're not competing for the same resource. So what that allows is that you can have a very big difference in fitnesses between these two populations. So maybe this one's very bad at laying eggs, this one's very good at laying eggs. That doesn't matter. They'll still coexist perfectly fine because you've got no competition between them. On the other hand, if you've got two species that look identical to each other, that eat the same food source, you're going to have a niche overlap of 1, and your fitness ratio has to be equal to 1 in order for those populations to coexist indefinitely. Because if one of them is able to grow more quickly than the other one, just with a higher fitness, then it's going to be able to, over long periods of time, take all of that resource and outcompete the other population. So that's the kind of qualitative picture. And again, you can understand this quantitatively within Lotka-Volterra. So basically, this is a result from Peter Chazon, where basically he showed that for a two-species Lotka-Volterra system, you have a fitness ratio, which is on this y-axis here, which is given by this expression here, and then you have this niche overlap, which is given by this combination of the interaction values over here. And that's actually what's plotted on these axes over here. So this coexistence criterion, where your fitness ratio lies between these two bounds, is what's represented by this hatched region over here. And if you're below that, then you're down here, and if you're above that, then you're up here. So one of the two populations winning. Remember that we've got this framework which allows us to translate any Lotka-Volterra expression into an equivalent expression which now holds for every environment organism system, just by exchanging our rates for accelerations. So that's what I've done down here. I've just put primes in front of all of these effective Lotka-Volterra parameters, and that's telling you that I've replaced those rates with growth accelerations in the relevant parameters. So this is very useful because it actually allows us to do something which people have not been able to do before, which is to explore mutualistic relationships between populations within this framework. So people usually think of this as a purely kind of, you're just thinking about resource competition, but now we can use this expression to explore how mutualisms map onto this kind of framework. So again, we've got the same model that we had before, where we've got our input media, so carbon and 2 amino acids. And now instead of just having carbon, I'm going to vary the concentrations of the input amino acids in this system. And then I'm going to ask, does one or both of the populations coexist in the chemostat over long periods of time? So that's what's represented on these two axes over here. So we have our input concentration of amino acid 1 and amino acid 2 on this axis over here. And we see that wherever I've put open circles, we have competitive exclusion in the chemostat, and wherever we have solid circles, we have coexistence of the two populations. And then I've also plotted these two axes over here, which basically say this is a constant ratio of our input amino acids, and this is a constant amount of our two when we add them together. So this is a constant total concentration, and this is a constant ratio of the concentrations of the two. We can use that term, that expression that I showed you on the previous slide, to map this onto these modern coexistence theory axes, and we see it works perfectly, which is not surprisingly, because this is just a kind of algebraic result, but it's still not nice that this works. So we see that wherever we end up mapping onto these coexistence boundaries, that is where we see competitive exclusion taking place in the chemostat. And we can relate this in various ways to the direction of motion that we have on these axes. So we see, for example, as we increase the total amount of amino acid but keep the ratio fixed, we're tending to increase the niche overlap, for example. Now we can do this a bit more generally. So we can now ask, okay, I'm not just going to change the input concentrations, but I'm also going to change the leakage rates of my amino acids. And I'm going to generate thousands of different simulations with combinations of these parameters, and then ask how what the average values of those parameters are in different regions of this coexistence space. So this is going to tell us how these interaction mechanisms are shaping coexistence effectively. So here, for example, we have our equilibrium nutrient concentrations. So this is our carbon. This is one of our amino acids. We see that when we have high levels of niche overlap, we have very depleted carbon, but lots of amino acids being present in the system. And down here, again, I'm not going to go through this in too much detail because I have a nice summary slide, but basically this is telling you how the variations in the input concentrations and the production rates are causing changes in these modern coexistence metrics. And to summarize what we're seeing here, basically when we look on the far right over here, what we've got is depleted carbon, but lots of amino acids. That's again this idea of variations in the relative dominance of amino acid competition versus carbon competition. So over here, we don't have very much carbon, but we've got a lot of amino acids. So our carbon is growth limiting, and both of these populations are competing for the carbon. So this is why we have a very high niche overlap, basically, is because both of these populations are competing for the same resources. Over here, however, the dominant driver of the dynamics is the amino acid concentrations, and these are orthogonal. So only one of the two populations can use both of these amino acids, right? So that's why we have very low niche overlap over here, is because we have independent regulation of the two populations by these two amino acids, which are determining the coexistence. Now over here, on this axis, now we can understand this fitness ratio as being down to variations in the relative flux of these amino acids. So up here, we see that there's a much higher flux of this amino acid to this population over here, whereas over here what we've got is a much higher flux of V to the corresponding autotroph. And so this is what is really driving the variations along the fitness ratio axis. Now that perhaps seems a little bit pie in the sky and a little bit theoretical, so I've got some nice results recently where we actually explored this experimentally. This is a slightly different model system, but it turns out that the same framework can be used. Basically what they did in this experiment was they had 15 gut species, and they had monoculture data for these 15 species. And what they did was they spent an awful lot of money trying to figure out exactly how each of these species modified their chemical environment. So they used metabolomics, basically. And remember that I said that you need to know 20,000 different chemical concentrations in order to effectively parameterize one of these models. Well, it turns out that's what they were able to do with this data. So they were able to generate a coarse-grained consumer resource, or EO model, and that model was parameterized using that metabolomic data. So you've got this monoculture data, which is highly mechanistic in nature, so we know exactly how the organisms are interacting with their environment. And we can then use this model in order to map onto these modern coexistence theory coordinates. And the key point here is that these coordinates are determined purely by the monoculture data. And then we're going to ask whether or not your position on these modern coexistence theory axes are predictive of where we are in terms of the pairwise coexistence outcome when we run a competition experiment over long periods of time. So we're using monoculture data in order to predict the coexistence outcomes in pairs. And we can show that this works basically perfectly. So on this blue line, we would expect to see the blue species winning, and on this red line, we would expect to see the red species winning. So this is a data slide, which I'm still slightly surprised it works this nicely, to put it bluntly, but basically what this is saying is that this kind of accelerational framework really is capturing something fundamental in terms of the ecology of these populations. So we can really use this to map onto coexistence outcomes. So now we've got basically a three-layered understanding of our system. We've got our mechanisms, then we've got the interactions in the middle, effective Lotka-Volterra parameters, and then we've got the coexistence outcomes at the top there. Okay, so it all kind of links together quite nicely. All right, to summarize, coexistence outcomes and growth in these mechanistic ecological models is second order. So it's not a first order system as you would expect for Lotka-Volterra. It's a second order accelerational process. Secondly, there are counterintuitive relationships between interaction mechanisms and effective Lotka-Volterra parameters. and this can give rise to resolutions to certain paradoxes that have been long-standing in the literature. And thirdly, when we take into account these second-order dynamics effectively, we can actually use this in order to accurately predict coexistence outcomes in these microbial ecosystems. All right, with that, I will thank the group in Switzerland, so Sarah Mitry and all co-workers. I was also funded by a fellowship up here, and this is a QR code that leads to my recent preprint, which I think is also being linked to on the website. So yes, take a look if you're interested. And then the first application that I talked about is already published, so you should be able to find that quite easily. All right, okay, thank you very much.